First, of course, I would like to say many thanks to Jerry Pollock, and how many? 12. For the 12th invitation to participate in these brilliant meetings. Though we have now the 11th conference on water in physics, chemistry, and biology, but all it started, if I'm not mistaken, in 2003, when there was the first and the last Gordon Conference on interfacial water, and because the organizers, the Gordon Conference Committee, decided that this topic is not very interesting. Probably they were mistaken, because after this, already 11 yearly conferences on physics, chemistry, and biology of water takes place, and I have had a privilege to participate, thanks to Jerry Pollock, in all of them. And this time I present a lecture. I had to present a lecture, something new even for me, though during the last conferences I was speaking a lot about water, reactive oxygen species, bioenergy, things like this, but my position is professor of the Department of Bioorganic Chemistry. And for the first time, I will concentrate on bioorganic molecules, substances. For the first time, I'll talk a little about peptides, about their importance. But being the conference on physics, chemistry, and biology of water, it is impossible, from my point of view, to consider what peptides are and what they are doing in all living systems, not taking into consideration water. First, I would like to start from the current status of peptides in biochemistry and bioorganic chemistry, to which I belong. In fact, if just to look into the standard biology textbook, you'll see that peptides are nothing very special. From this citation from the standard biology textbook, where it is written that peptides are distinguished from proteins on the basis of size, as an arbitrary benchmark can be understood to contain approximately 50 or fewer amino acids. So we just we agreed to consider that if some polypeptide contains more than 50 amino acids, we call it protein. If less than amino acids, we call it peptide. Some people could think about 40, others could think about 60, but nevertheless, nothing very special about peptide from point of view of bioorganic chemistry. Though there is also the word oligopeptides. These are peptides from 2 amino acids which combine together to about 20. These are small peptides which are called oligopeptides. So that is what we give our students, and usually we consider peptides as small, very small proteins. Now, how these peptides and why they may be important for biological processes. Where from they appear, these peptides? So they appear, most of these peptides, they appear due to normal protein cleavage by proteases. Why, when proteins are already used, they are broken, oxidized, something happened to them, they wear out. Then proteases begin to cleave these proteins into smaller parts, which we call peptides, according to the definition. And then these peptides are cleaved by peptidases into free amino acids. So from this description of appearance of peptides, again, nothing very special can be seen, though there is another way of origination of peptides in living systems, such as, for example, bacteria and fungi. When peptides are synthesized, not using specific enzymes, which consequently add one amino acid to another, to the third, to the fourth, and in fungi, bacteria, there appear such sometimes very exotic peptides which have antibiotic functions like gramicidin, braccitracin, or antamanide, and other interesting molecules. And in fact, there is a third way of peptide origination, which I skipped for not to overload my presentation. This third way of peptide origination was discovered in the 1930s by Alexander Gurevich, who was the discoverer of mitogenetic radiation. I'll tell a few words about this mitogenetic radiation later on a little. So he discovered that if you have a solution of amino acids, for example, the simplest amino acid, which is glycine, and you irradiate it with a very weak flux of ultraviolet photons, the process starts in this solution of glycine and other amino acids, in which the polycondensation of amino acids occur, and they convert into peptides and polypeptides. And this is an autocatalytic reaction. So this is the third way of peptide origination. Very, very few people know about this. We reproduced some Gurevich experiments about 20 years ago and have shown, yes, that peptides may originate this way. So even these two examples, last examples, this one and examples of Gurevich, show that peptides may be something else than just the byproducts of protein cleavage. And I just pose at the beginning of my talk the question, could it be that peptides are not just small proteins, as it is written in biological textbooks, but rather they represent a specific class of biomolecules? So I will talk about this. Now, already it is well known for several decades that these peptides, which originate even from protein cleavage or some other way, can possess a very wide spectrum of biological activity. I simply talk about things which should be known to every biology or biochemistry or physiology student. That in nervous systems, peptides play a role of neurohormones, neuromediators, neuromodulators. No less important, and sometimes, as we will see a little later, more important than low molecular weight neuromediators such as acetylcholine or serotonin and so on. In fact, everybody knows such words like enkephalin or endorphin, which produced in our body, and morphine and other drugs imitate their action, which is in fact normal action. In digestive system, peptides are key factors, play the role of major hormones controlling various functions of digestive organs. Just to name gastrin, secretin, glucagon, and so on. In immune system, peptides are key factors mobilizing defense of an organism against foreign invaders, so they work something like antibiotics or endotoxins which are produced in our body. In cardiovascular system, it was discovered not so far ago. There are peptides which are called natriuretic peptides. They promote blood pressure reduction, play a role after thrombotic agents, and besides exhibit anti-lipolytic activity. So the widest spectrum of signaling activity, but usually people again do not differentiate these peptides from low molecular weight signaling molecules or high molecular weight protein hormones, for example. So they look at peptides in this row. But only recently, many biologists, physiologists, biochemists begin to recognize that peptides may play a key role in one of the most important biological processes. I would say the intrinsic biological process, the process of development of tissue and organ development, and also one of the most important processes, that is regeneration of impaired organs and systems in all eukaryotic kingdoms. Here I give a very few examples of this role of peptides in development and regeneration in all eukaryotic kingdoms. The unicellular eukaryotic cells like yeast, they mate, and their mating is guided by mating pheromones. Oligopeptides having 12, 13 amino acid residues. Very impressive effect of peptides, of so-called neuron regeneration peptides, which stimulate neural proliferation, migration, and differentiation with no apparent toxicity and high target specificity in central nervous system. As a matter of fact, I'm afraid that some of you still believe in school knowledge that neurons simply disappear and they cannot multiply and so on. Now it turned out that it is wrong. There may be regeneration of nervous system in principle even after very severe seizures in brain. And these peptides, they support this process. And this understanding that peptides play a key role in development and regeneration, it came from another kingdom. It came from understanding that peptides are very active in plant kingdom. Because it was discovered only 15 years ago, maybe some 20 years ago, that plants contain numerous peptides which control practically all developmental processes in plants, from fertilization, seed formation, to cell proliferation, differentiation, and abscission. Of course, people study peptides in many, many different plants, but there is, among all the plants, there is the beloved model plant, which is called Arabidopsis Thaliana, a very small higher plant, which plays the role of Drosophila in plant physiology, plant genetics, plant biochemistry, and so on. And for this Arabidopsis thaliana, whose genome is decoded completely for a long time and studied very well, from this genome it is deduced that at least 100 signaling peptides should be produced by proteins which are cleaved during their lifetime in plants. Only few of these peptides are very deeply studied. And one of these peptides, so for example, so here are the indexes, the names of this peptide. And one of these peptides which is studied best, which is called peptide CLV3, it originates from the protein CLV3, is the best studied peptide because its receptor is also found, a receptor like kinase CLV1. So this pair modulates stem maintenance and cellular differentiation in shoot meristems. It is these two proteins are expressed in shoot, and they are important for growth and differentiation of stems. But recently it has been shown that if to take either extracted from plants this peptide PCLV3, which is here is in one code language, 12 amino acids are contained in this peptide. If to add either synthetic peptide PCLV3 to culture medium of Arabidopsis, then it would influence root growth, though no putative receptors of this peptide were identified in roots. And we, being at the Department of Bioorganic Chemistry, using the synthetic PClV3 and some of its analogs, of which a little later I'll say, we decided to study how it really works if to add this peptide into the I'm sorry. If to add this peptide into the culture medium of these small Arabidopsis plants. And being the follower of those people in Russia and other countries who study ultra-low doses, what was really interesting for me to study ultra-low doses of this peptide, because they, in real organisms, they are present in very, very small concentrations. So we begin to study effects of this synthetic peptide, ClV3, in concentration, including ultra-low ones, on the main root and on lateral roots growth of this Arabidopsis. Now, main root growth, and I will dwell on this a little later, is growth due to proliferation and elongation of cells. That is just cell division. And appearance of lateral roots. It's the sign of differentiation. So it turned out that in comparison to control, to which this peptide was not added, in very wide range of concentration, here is 10 to the minus 12 molar calculated concentration. We studied it to 10 to the minus 16 molar. It produces very complex effects on of this peptide on both processes, process of root growth and process of root differentiation. So here is the plot of concentration dependence of the effect of this peptide on the length of this peptide is blue, and two other curves are its homologs, which are not from Arabidopsis, but from completely another plant, which is moss, the lower plant. And still they have similar, not identical primary structure, and they produce a similar effect upon this plant. This affects biphasic or even three-phasic effect. In concentration range from 10 to the minus 6 rather to 10 to the minus 8 molar, you see a very profound inhibition of a major root growth. When concentration decreases or with further dilutions, effect disappears, and usually people stop here. There is no effect. But if to go further with this concentration, then one can see that here there is even stimulation of root growth. And then when you go further, there is a more modest, but still again it appears the retardation of root growth. So what decrease in root growth is just a kind of poisoning effect on plants. In fact, no, it's not the case. If to look at the same plants and the same roots, how in this concentration range this peptide affects the appearance of lateral roots, you can see the difference here is 1, and here each plant has many, many different roots. One can see that in this concentration range, when it retards the major root growth, it stimulates tremendously the lateral root growth. Then when we go to lower concentration, there appears another pattern of action of this same peptide molecule in concentration around minus 12 and even more. There again increase in lateral root growth, one can see it here, and stimulation, continuous stimulation of major root growth. So here it's already interesting thing that very, very low concentrations, in fact, there are practically no real molecules in this in the culture medium of the cells. This peptide, or in fact, why not to say water, which is modified with peptide, produces different pattern effect upon this plant, its root growth, than in a higher concentration range. Now, so we have here effect of one and the same stuff in different concentration ranges, which have two different patterns. We were looking on two processes which are related to the development of the growing plant. Cell proliferation and cell differentiation is the key aspect of development in multicellular organisms. Cell may multiply and then they differentiate. And then they again may multiply and then again differentiate. So multiplication and differentiation, these are two processes which define the pattern of development of multicellular organism. And it was discovered very, very recently that this balance in Arabidopsis roots, it was not discovered, it was proved, is modulated by the balance of reactive oxygen species between the zones of cell proliferation and the zone of cell elongation, where differentiation begins. So this paper was published in very respectable journal Cell only 6 years ago, where it was shown such interesting thing. First, I would like to remind you what are reactive oxygen species. These are intermediate products of oxygen reduction by electrons plus protons. The first one reactive oxygen species is superoxide anion radical, when oxygen molecule gains one additional electron and converts into what we call free radical. Another reactive oxygen species is hydrogen peroxide, which appears from recombination of two superoxide radicals. And then hydrogen peroxide may give birth or may not give birth to hydroxyl radical and so on. As probably most of you heard, some of you of course know it very well, reactive oxygen species until now considered to be a very harmful byproducts of normal metabolism. But what role they play in proliferation and differentiation of these roots? So it was demonstrated that the root consists practically of two parts. There is the lower part of the root where cell division or proliferation takes place, and there is the upper part of the root. There is a so-called transition zone between them where cells begin to differentiate and give, like the products of differentiation, lateral roots. It was discovered that superoxide radical is abundant in the lower part where cells divide, and product of their recombination, hydrogen peroxide, is abundant in the upper part of the root where differentiation goes on. If by some means or in special mutants to produce effect when the production of superoxide radical is retarded, it becomes much less intense, then hydrogen peroxide concentration increases and roots become small, but differentiation becomes more pronounced. If, on the opposite, to have a mutant or by some other chemical or other means stimulate production of superoxide radical and retard the diminished concentration of hydrogen peroxide, then root become to grow much more intense. So proliferation in these cells, in cells of meristem, is stimulated, and differentiation is retarded to some moment. So in order to understand if peptides added to this these plants may somehow affect the production and generation of reactive oxygen species, we turned to the basic biological, to understanding and to considering the major biological function. That is cell division, because cell proliferation or retardation of cell proliferation, of course, depend in the first place on cell division, on mitosis. So the mitosis of cell is the major process promoting cell growth. And there are two alternative mechanisms of mitosis triggering are suggested. One mechanism, which is very well known and which is only considered in biological textbooks, is the mechanism based on receptor mitotic hormone interaction. It is considered that all cells contain specific receptors, and there are hormones which produce their division. When hormone binds to the receptor, it is a signal for triggering of mitotic process and cell divide. But again, Alexander Gurevich, whom I already mentioned, in 1920s, he found that mitosis may be induced by the so-called resonant factor, physical factor. Here is the resonance model. He considered that cells which are ready for mitosis are in an excited state, and there is a physical stimulus, for example, photon, which hits the cell surface, and cell is initiated to go into mitosis. And Gurevich proved this model, and for many, many decades, people were really pretty sure that mitosis may be induced by physical factors. As a matter of fact, at the beginning, as a model, he was used roots of plants. Here is onion plant. So here is the root, which is the source of mitogenetic radiation. It is targeted on another root where cells are ready to go to mitosis, and it was discovered that those side of the root which is enshrined by this low intensity level of photon emission, on this side the quantity of mitosis is statistically much higher than on the shadow side. So Gurevich experimentally proved that mitoses are triggered with ultraviolet photons emitted from tips of onion roots. Now, where from these ultraviolet photons originate in living systems? It's not only roots, it's bacteria, it is fungi, it is yeasts, and all other mitoses and all other living cells are triggered by mitogenetic radiation. So the source of this mitogenetic radiation, as it turned out, is the recombination of free radicals, starting from superoxide radical and other radicals. When free radicals recombine, there is a very high quantity of energy is released in the form of a photon belonging to the ultraviolet part of the spectrum. So when this photon hits the cell, which is ready to divide, the cell goes to division. So we now already combined cell proliferation, which depends on mitogenetic radiation, and mitogenetic radiation is dependent upon the recombination of free radicals, which originate continuously in all living systems. So the question was, do these peptides affect the metabolic processes involving or turnover of different free radicals in biological systems? And we have such a model biological system, which is just usual normal water, bicarbonate solution. So we long ago demonstrated that in bicarbonate aqueous solution, just drinking water which is standing on your table, there is continuous production of superoxide free radicals. And we suppose, and I presented our consideration of this question, that these free radicals in normal drinking water, they originate due to continuous coexistence in these two different water phases. One phase is kind of easy water of Jerry Pollock, and another phase is bulk water, in which different substances are dissolved, including oxygen. Easy water, as we now know very well, is negatively charged, so it has excess of electrons which may be released, and released electrons will combine with oxygen, and they will reduce oxygen, and on the way of oxygen reduction back to water, there appear reactive oxygen species. And this appearance of reactive oxygen species can be monitored, because, as I said, their recombination is the source of photons. And these photons, which originate due to their recombination, may be monitored using photomultipliers. For example, if you take bicarbonate aqueous solution and add to this solution, for example, the salt in catalytic quantities of reduced iron, this iron is oxidized, adds additional electrons to water. Electrons in water begin to recombine intensively, and we have a photon flash. So intensity of photon emission from water to which we, bicarbonate water to which we added this reagent, we call it reagent, is related to the level of superoxide radical in this solution. So the higher is the photon emission wave, the highest concentration of superoxide radicals or stationary state. The lower this stationary state. So the question was, can small signaling peptides affect the balance of reactive oxygen species between the zones of cell proliferation and the zone of cell elongation where differentiation begins? And we tested if peptides that inhibit root growth can affect generation of ROS in much more simple aqueous system in bicarbonate aqueous solutions. So we studied again the concentration dependence of this peptide upon intensity of photon emission from bicarbonate aqueous solution. And you see that in concentration range from 10 to the minus, up to 10 to the minus 8 or even 10 to the minus 10, there is inhibition of photon emission from this solution. Then this inhibition disappears, and then it appears again. And here I wrote the concentration dependence of the effect of this peptide on root length. You can see that these two curves are very, very similar to each other. So from this, at least, we can make a preliminary statement that the peptide somehow inhibits production of superoxide radical, and due to this inhibition of superoxide radical, it inhibits the root growth. Where inhibition is eased, then root begin to grow more intensively, and so on. Now, this why peptide produces so many different biological functions, including regulation of developmental processes, and as a consequence, regeneration of impaired biological function in specialized organs and tissues. And can these ubiquitous functions of small signaling peptides relate to their ability to modulate oxygen-dependent processes in aqueous system? So we come to the question which was asked at the very beginning. What is so special about small signaling peptides from the point of view of physics, chemistry, and biology of water? And now I turn to the theory, one of the theories which in total built the theory of association induction of Gilbert Lin. This is polarized oriented multilayer theory of cell water. According to Gilbert Ling, proteins which do not make internal hydrogen bonds building alpha helices and beta sheets, which are in extended conformation. He wrote that a backbone of peptide bonds, backbone amine and carbonyl groups, are the primary seat of water sorption. But especially the carbonyl atoms with their lone pair of electrons. Now, what is the difference between peptides and proteins? Most of peptides may be looked upon as fully or at least extensively extended molecules, and their peptide bonds are open to interaction with water. And so that is the major similarity between different peptides. Regularity of hydration of the common feature of all peptides. But all peptides have different amino acid contents and amino acid sequences. And this is specificity, which is a unique feature of a particular peptides. So we come to the hypothesis that probably these two features of small signaloid proteins, peptides' ability to form multiple layers of highly polarized water, and specific hydration of particular amino acid residues may provide for their role in regulation of multiple biological functions, because they will affect the major bioenergetic and bioinformation process which goes on in living system, that is the metabolism of reactive oxygen species. And now I come coming to the end. Probably I have 1 minute and it will be enough for me. So it is interesting to speculate that due to limited number of building blocks, amino acid residues, patterns of hydration such molecules, bioorganic molecules as peptides, should be significantly different from those characteristic for those small molecular weight regulatory compounds and big molecular weight regulatory compounds. And I'll finish with the, I even don't call it hypothesis, but probably at this point it is kind of fantasy. Taking into consideration that peptides with necessity originate at the final stage of any protein life cycle. And what is protein life cycle? Protein, again, it is split into peptides, and then peptides, they cleaved into amino acids, and new proteins are built from these amino acids. So one may imagine that not only protein emergence, but its elimination through peptides may be a stimulus for rejuvenation of the living systems. So peptides which originate during any protein degradation to free amino acids, they should be kind of a stimulus for signaling, regeneration, and rejuvenation of a living system. This hypothesis, or fantasy, I would say, it is very close to some ideas about the process of apoptosis. Some people think that apoptosis is the so-called programmed death of cells, maybe not very good for living organism. But I would say that the major, most intense apoptosis goes at the stage of embryo development. About 90% of nervous cells go into apoptosis, and about 99% of gonad cells go into apoptosis. And without this apoptosis, without death, there is no development, there is no origination of the new life. So, in few words, again, the final conclusion, peptides which with necessity are the products of death are the stimulus for future life. And I hope that this hypothesis is crazy enough for pretending to become true. So it's just after Niels Bohr. Thank you very much.
Thank you very much, Vladimir. Thank you for your wonderful presentation. And it was, very nice, wonderful that even the differentiation process in plants can be related to the property of water. And you have any questions? Maybe 2 questions, because of the time is limited. Over there?
Thank you, Vladimir. This was quite exciting. Using the association induction hypothesis on Gilbert Ling and with your peptides, and referring it to the hydration that you just depicted on the screen, could I imagine that I use an electromagnetic signal triggering off a chemical reaction in such a way that I use the peptide, the hydrated peptide, as the interface?
It's very far. You are very far from here.
Sorry. To make it concise, okay, can I use the hydrated peptide as an electromagnetic interface in order to trigger off a biochemical reaction?
Mm-hmm. Yes, and it has been demonstrated. You see, what is really very interesting, that peptides have a versatile, a wider spectrum of biological activities also. And just in previous talk, it was said that some peptides may interact not through receptors, but bind to cell surface. I believe, together with Jerry Pollock, that cell surface is covered with easy water, and peptides find their place in this easy water, being also hydrated, covered with some analog of easy water.
Thank you. One more question? Okay.
The presenter for his great work in generating Gurwich's influence. I know at the Biodesign Institute in Arizona, a lot of work has been in the sound rejuvenation of plants through the piezoelectric effect in the root structure, and a lot of work is being done in Indonesia and China now on some extensions of Gurwich's original hypothesis.
I would just, as a remark to your remark, I would like to add one thing which may be interesting and important for all the present here. Peptides now appear as a completely new class of pharmacological agents. This class is very different from low molecular weight compounds, which are synthetic, or from such things as antibodies for immunization, like proteins. Because these peptides are natural, practically always natural, and now they begin to understand more and more that they induce the process of regeneration, which is most important process. It's not curing, it is healing, when the lost function is returned back under the action of peptides, as a matter of fact, in very, very low concentrations.
Thank you very much. Yeah, so we would like to move to the next session. Thank you.